Hello, good afternoon, and welcome to the last and final sequel of our lecture. I think I have had two classes, on-site classes with you already, and I chose it's better to conclude this lecture, even though it's going to be virtual, but I hope the information is going to be passed across, okay? By and large, you have the course material already, right? Forensic biotechnology and application and its application. Forensic biotechnology application, right? So, done most of these slides, I'm sure. And I think we are supposed to continue from southern blotting. Okay. All right. Southern blot analysis. Okay. There's northern blotting, there's western blotting. Western blotting is done for protein samples. Northern blotting is done for RNA, while southern blotting is done for DNA. The southern blot analysis reveals information about DNA identity, size, and abundance. There's a classic technique that involves separating DNA fragments based on size via electrophoresis, which we've done in the previous class, which we've discussed, of course, transferring them to a membrane hybridization with a labeled sequence-specific probe, washing, and finally detection of labeled DNA. So all this will be explained in the next slide, to be honest. So what's done here is also done for proteins. If it's a protein sample, it's called western blotting. If it's RNA, it's called northern blotting, or this is DNA. So extract purified DNA from a biological sample. Of course, you are aware of techniques used in DNA extraction. And then we digest the DNA using restriction enzymes. In the last class, I discussed restriction endonucleases, right? These enzymes digest DNA at recognition sites. Equal R1 has a different recognition site to band HI. For example, these are two types of restriction enzymes. So after using a restriction enzyme to digest the DNA, and you separate the nucleic acids by size using gel electrophoresis, which we also discussed in the last class. And then you denature the DNA, denaturation, right? After denaturing the DNA, you apply nitrocellulose membrane to the agarose gel so you can capture the bands, right? So blotting and migration of nucleic acids onto the membrane. This is the technique. You have a sponge, you have the agarose gel, then you have the nitrocellulose membrane top. Using UV light, you can view your bands. Yeah. So this is your nitrocellulose membrane with bound but unlabeled nucleic acids. It's showing nucleic acid, but they are not labeled. So you must be interested in particular nucleic acids, and that's why you have to hybridize the nitrocellulose membrane. And you're hybridizing the nitrocellulose membrane with probes, sequences that are complementary to the DNA fragments you're interested in. And then those probes have an indicator, a color or something that will show that they've attached to your fragments of interest. And that's how we are able to visualize those DNA of interest. Without the probe, you can't identify your DNA of interest on the nitrocellulose membrane, right? So this word is just English, really. This is in English. Don't get confused. You are being probed. An investigation is going on. You use something to catch something, right? So this probe is like something you want to use so I can catch that DNA fragment you're interested in. So you can tackle it, can identify it using the biochemical term, right? Let me not talk like a layman. All right. So the genetic detective, all right. The Golden State Killer, that's I think in America, I think or UK, I'm not sure where, but there's a serial killer, they gave a nickname of the Golden State Killer, was found using familial DNA analysis. So family members uploaded sequences to genetic genealogy sites online, right? Forensic scientists uploaded crime scene DNA to identify familial connections. So because they were family members of that killer who have uploaded their DNA information online, they were able to uh, zero in on the killer because they had the DNA material from crime scene that was not of the victim, but of the killer. And if they have already family members who have similar, not identical, similar DNA sequences, you see it narrows down the search for police detectives, right? They know that the killer is within this family. Now they can zero in, look for members of the family, look for warrants, and look to how to identify those members of the family who fit the description of their killer. Is it a male or a female? And whose DNA information is not available online, right? Using common sense. Yeah. So topic for discussion, is it ethical to use this information to identify potential suspects? Of course, that's the next question. There's the issue of ethical breach, right? Breach of privacy. Your family members have the right to privacy if you upload your DNA information. Of course, we discussed this in our first class, I think, right? So how do you answer this question? Anyway, that's up to you. Electrophoresis separates DNA fragments by size, right? We already know this. Samples are loaded into the wells and an electrical current is passed through the gel. I gave you a YouTube link to watch, right? So it's the same thing here. The sugar phosphate backbone of DNA has a strong negative charge. Of course, phosphate has a negative charge. The current drives the DNA through the gel towards the positive electrode. Small DNA fragments move through the pores easily because they are small, right? Or large DNA fragments have a more difficult time squeezing through the tunnels. Of course, it makes sense, right? The DNA separates into distinct zones based on size, of course. All right. Okay. So I think so. Usually DNA is clear and colorless. Well, this color here you are seeing is due to dyes or stain we use so we can see the DNA bands, right? And if you've watched those videos I sent to you, then I think you know this. But in conclusion, a match provides strong evidence that the suspect was present at the crime scene. That's when you do DNA extraction, amplification, and sequencing. Alone, this evidence does not prove the suspect committed the crime. If the suspect DNA profile does not match that of the crime scene, the person may be eliminated from the inquiry. Of course, we know this. We can read this on the application of DNA forensics. I've given you the material already. I assume you've read this already, actually. And then you have this. This is just more like a summary, right? Okay, forensic analysis, next generation sequencing, ancestry. uh, Sequencing is key to this aspect of genetic engineering, right? This area of genetic engineering. Depending on what type of sequencing you want to do, but sequencing is very important here. Wouldn't you agree? Okay. If you remember the short tandem repeats, the SNPs, when we were doing the restriction fragment length polymorphism, when I was talking about how restriction enzymes work in the last class. I hope you still remember, right? Those with biochemistry background should know what microRNA is to be used for potential candidates for modified identification. If you don't know, you can read about it. Everything is available online. Okay. Yeah, yeah, editing. I think that should be real time PCR. Okay. STR genotype in short time then repeats. It's just take your time. Take your time. take a look at this. Summarizes a lot of things. You get an idea of what we're talking about. QIE, quick spin column purification. This is like DNA kits used for extracting DNA. Something like this. It's a company. You can type this name online. I'm sure the company will come up. This should be Kaijin, I'm suspecting. All right. Lab on chip nanotechnology. This is very interesting. It's very interesting. Okay. All right. Metabolomics. So you want to determine if from autopsy the patient was poisoned or something, and to check the metabolites. What are the normal metabolites for a healthy individual or a dead body that was not poisoned compared to a one that was quite poisoned, for example, metabolite and things like that, right? So you have techniques, extraction techniques, column chromatography, right? Mass spectrometry, things like that, LCMS and all that. So all these techniques work together. This will be the end of our lecture. Thank you for listening. You can talk to the second lecturer. I mentioned that. And for continuous assessment, reach out to your class rep. We'll see how to handle that. If you have any question, you can leave it in the comments. And don't forget to like and subscribe to our channel. Thank you very much.